Okay, I actually have to admit I slightly changed the title of my talk because when I started preparing it I realised actually that I wanted to talk about two things really in the research that we've done in the years gone by, which deal actually not with long distance navigation, which is what I was going to talk about exclusively, but even medium distance or medium range navigation in nocturnal insects. So I sort of slipped into navigation almost by accident because my actual real area of interest, and I feel like a bit of an imposter actually being an organiser of this course. My real interest is in the neural and optical strategies animals have evolved to see well in very dim light, both at night and in the deep sea. So that's my actual interest in terms of research. But of course, animals that need to see well in dim light need to do so in order to do things. And one of the things that is surprising is that many nocturnal animals, insects included, turn out to be really remarkable navigators actually. And so what I want to tell you a little bit about is a couple of case studies where we've explored this in more detail. One of which is medium range and one of which is long range in nocturnal insects. So already in the first lecture and in other lectures we've heard during the course, we've talked a little bit about insects being able to navigate in bright daylight. And in bright daylight, especially if you're using vision, then navigation is usually quite sophisticated and reasonably straightforward because you've got plenty of light to do it. And we already talked about this, I'm not going to go into this in any detail, we talked about Kathyglyphis as being a fantastic example of a mid-range or medium-range navigator, and indeed many of you here in the room work on this and still for that reason. And we talked about the fact that they had path integration that required a compass, which was celestial polarized light, and an odometer, which in their case was an idiothetic step counter mechanism, and they're really excellent navigators. They're also able to use various kinds of sensory landmarks, you might recall, both visual and olfactory, to find their way back to the nest after a long foraging trip. And there are plenty of examples of excellent day-active navigators, both insects and non-insects, vertebrates, and which rely heavily on vision. And visual cues, visual landmarks, visual other celestial cues like the sun are very, very obvious to day-active animals, and they're used very heavily for navigation. We also mentioned many times during the course so far this remarkable insect, which is a long-range navigator, unlike Kathyglyphis, which is a medium-range navigator. And as you might recall, this amazing animal is able to migrate thousands of kilometres from northern parts of the United States, southern parts of Canada, and migrate thousands of kilometres to a very specific location in Mexico, and then find their way back again. And they use the sun compass, you might remember, to do this. The sun being an extremely bright, a very constant, a very predictable, and very reliable cue, day in and day out. It's a great cue. Turns out that nocturnal insects are also really phenomenal navigators, and there's a rather large number of cues that they could use. Obviously still visual. I mean, as Henrik pointed out, some people actually think there's no light at night, which is clearly not true. There's plenty of light at night, and you only have to satisfy yourself about that. Going far away from a city on a starlit night, even on a starlit night, which is pretty dark, we can still see things. We don't see very well, but we still are able to see something. But apart from just the regular light, there are other cues as well which are visual. The disk of the moon, for instance, is a possible cue, a navigational cue, like the sun is for monarch butterflies, for instance. The polarized light pattern formed around the moon, which I talked about earlier in an earlier lecture, and Marie Ducker also mentioned, is another possible navigational cue that's visual. Then, of course, we have stars. And then we have all the terrestrial objects around you, or which animals have around them, which are illuminated by this very dim light from stars and the moon. They're also possible visual cues. Of course, olfactory landmarks are used even in daytime. We've heard that already. Anna was very clear on that with birds. They're very able they use olfactory cues in navigation. And this is also true of insects. And there are also magnetic cues which are possible. Earth's magnetic field is an excellent cue for, especially at night, as we've already heard this morning from Peter and Henrik. And finally, there may even be some information that's mechanical in the case of various types of winds, for instance, which might be possible cues for navigation. And I'm not going to go into all of these, but olfaction obviously is a realm that is, for many insects, a very well-developed realm. And many insects have exquisite olfaction, and many of them nocturnal insects use olfaction for medium-range navigational tasks at night. But I'm not going to talk about that. Because what I'm interested in, being a vision researcher at heart, is to look more at the idea of how animals might use their visual systems at night for navigation. And this, it turns out, is severely challenging. And it's challenging for the obvious reasons that you would think of. And that is that at night time it's very dark and it's not very easy to use visual information to do complex tasks like navigation. So it's a challenge. And the reason why it's a challenge, and I'm just going to give you a little bit of information about what the world of light is like for animals, is that, if you go out into a field somewhere far away from a city and stand there for 24 hours and it's brilliantly sunny during the day and it's a starlit moonless night at night, then you'll experience a change of light intensity that is equivalent to about 100 million times change. So that's 8 orders of magnitude difference between a bright sunny meadow and a starlit meadow. That's a huge range of light levels. And not surprisingly, very few, if any, animals are actually well adapted to all of those light intensities. Animals tend to specialise. We have evolved to be day active animals, so we see quite well at the brighter end of that range, but we're not real good at the dimmer end. If you go under a rainforest canopy, or a dense forest canopy at least, light levels can be a further 100 times dimmer. So becoming extremely dim under a canopy, and I think Henrik even mentioned that in these environments it can become very dark indeed. But it's certainly not lightless. There's plenty of light there nonetheless. It's just very dim light. And the obvious problem for any visual task, including navigation in very dim light, is that vision simply becomes unreliable. And the reason for that is fairly obvious. That is that the light-sensitive cells in the retina of animal eyes, the photoreceptors, they have greater and greater difficulty in absorbing sufficient light to create a visual signal, a visual impression. At the same time, the noise in the photoreceptors, the actual sort of unreliability, if you like, in the signalling within the photoreceptors, proportionately at least, becomes larger as the signal drops. So in other words, the signal-to-noise ratio falls as light levels fall. And this is an enormous challenge for animals that live in dim light. For insects, it's particularly challenging. And the reason for that is that insects tend to have very small eyes and very small visual systems, and very small brains. The compound eye of insects, unlike our own eye, you can place most compound eyes of insects through your pupil, so small they are. And that means immediately that they are likely to have a poor ability to capture enough light to see well in very dim light. Particularly animals whose compound eyes are adapted to day vision, like classic day-active insects, like bees, flies, dragonflies. Many of these insects are unable to see at all in dim light. But I'm going to give you a case study in a moment of an animal.
What is that?
I'm sorry?
What is that?
Oh, that's a honeybee. Sorry. A honeybee head. Yeah, a drone, actually, I think. I'm not sure if it's actually Apis mellifera, but it is definitely a bee. It turns out that over many years of research in our lab at least, and in others now as well, it turns out that nocturnal insects have truly remarkable vision. Much better than we do, I would wager, in very dim light. That moth, for instance, up in the top right-hand corner, which I'll come to later, that's the elephant hawk moth, a beautiful European hawk moth that hovers in front of flowers like a hummingbird and sucks nectar on the wing in the dead of night when we're fast asleep. This animal we showed about 15 years ago has full trichromatic colour vision, just like we do, except it's in the UV, blue and green range of wavelengths, rather than the blue, green and red range of wavelengths like we have. And they use this ability to find flowers at night. This was the first animal known that was nocturnal to have colour vision. There's been a few since then that have been identified, including that bee actually in the middle, a huge Indian carpenter bee, which also has trichromatic colour vision at night, also for the same purpose, for finding flowers at night. And Marie already talked about the visual abilities of dung beetles at night, and there are also many different species of nocturnal ants that have really quite remarkable navigational abilities at night time. So what I want to tell you about in my lecture is the stories of two really quite remarkable and quite different nocturnal navigators. One, it turns out, this one on the left here, Megaloptogenalis. This is a Central American sweat bee which we've been working with now for almost 20 years. It lives in Central America. I'll come back to it in a moment. It's a strictly nocturnal bee. This is the bee that Stanley mentioned yesterday. He's done so much of his work on. It's a medium-range navigator, it turns out, in light conditions which are still, for me, so unbelievably low that I still don't fully think that we understand how it's able to achieve what it achieves. We've got some good ideas, which I'll get to in a moment, but honestly, I don't think we've got to the bottom of this yet. The other is this creature. This is the Australian bogan moth, Agrotis infusa. This is a long-distance navigator. This is the nocturnal monarch butterfly. There are only two species that do something like monarch butterflies, that is, that fly from many different origins and arrive at a very restricted location and then stay there for some time and then return. This does exactly the same job in Australia, and I'll come back to this later, and it travels over about 1,000, 1,200 kilometres to a very restricted location, and then the same individuals 3 months later return to where they came from. And I'll come back to this animal a little later. This is a long-range navigator, a nocturnal navigator. So I'll start with this wonderful bee, Megaloptogenalis, the Central American sweat bee. This bee has really very well-developed eyes. This is a sort of a falsely coloured electron micrograph. The three funny-looking eyes in the middle of the head are known as ocelli. We don't know exactly what they do in this bee, and we haven't got a single role for these in any insect, actually, but they, among other things, seem to be involved in flight control. But the compound eyes are well-developed, just as well-developed, in fact, as any day-active bee. And the work that we've done on this bee has been at this place. I don't know how many of you have ever heard of this island in the middle of the Panama Canal. Definitely Narcom, of course, yeah. Barro Colorado Island. It's probably one of the most famous tropical research stations in the world, run by the Smithsonian Institute. It used to be the top of a mountain before they made the Panama Canal. And the Smithsonian Institute have a really excellent field station there. Those of you who work on bats, this is a great place for bats. Yeah, Malcolm is nodding his head there. So the bees that we've been working on in this rainforest here just beside the field station. And as Stanley already showed you, this is the world of this bee. This is what the bee has to face every day. And if you go walking through here as a human being, just walking along the tracks of this rainforest, and if you stray off this track, it's very, very likely you'll get lost pretty fast if you don't find your way back again. It's extraordinarily difficult for a human being to navigate their way through that mess. These bees I'm about to tell you about, though, do this every single night, in the dead of night, without any trouble at all. And we already know from pollen samples collected from these bees that they travel at least half a kilometre away, at least, probably further, and find their way back to this. A very small, hollowed out stick hanging in the undergrowth. This stick is typically about that sort of length. It's just a stick that's fallen down from the canopy. The bee has hollowed out a long chamber in the middle of this stick and has cells on each side of the chamber where they raise their young. There's usually only one bee per stick, but you can sometimes find more, and this is a female. And they leave this stick in the evening and fly off into the rainforest to look for flowers, and then they find their way back again under the rainforest canopy, we presume, in extraordinarily low light conditions, with a visual system which, at least at first glance, wouldn't seem capable of actually subserving such a task. So what I'm going to show you here is a film of 6 of these nest sticks on a stand in the rainforest. Now, as is sometimes the case, you do an experiment that doesn't work out. So each of these nest sticks, and if I can get this pointer to work. Yeah, so you see the 6 nest sticks here, and there are half ping pong balls attached to the end of them. I can explain what that experiment was about if you're interested later, but it was a complete failure. But it doesn't matter because the film demonstrates something else which I want you to see. Okay, these nests are all unoccupied except for that one. There's a single bee in that nest. Okay, this is all taken with a camera capable of detecting infrared light. This is all in infrared illumination, and it's at quite a dim light level. And there is a bee that will emerge from here to go on a foraging trip. And what I want you to notice is that the bee will come out, it will turn around in midair, and it will begin to survey the landmarks around its own nest, apparently, visually. And this is known as an orientation flight, and it's very, very well known in day-active bees, and it's used precisely for the purpose of understanding the arrangement of landmarks around the nest entrance so that the bee can remember them upon return. So if you look here now, let's turn that sound down. If you look here now, you'll see the bee emerging, okay, and then it'll start to fly, and it seems to be looking at things. Now you can't see it anymore. If you're really eagle-eyed, you'll see it zoom back through the film again. I've failed to see this every time I've played it in the last 10 talks. I still didn't see it, but I don't know if anybody did. Did anybody see the bee come zooming back through? No, it did. So the bee has sort of come out, faced the nest, and begins to survey the arrangement of landmarks as it makes ever-increasing backward arcs in flight before it then takes off and flies away. And so this looked for all the world to be exactly the same type of behaviour you'd see in a day-active bee, suggesting that they're using vision to learn. Yes?
I have just a quick question about this bee. So you said she, so it's usually a female that does this?
Yes, it is.
So what does the male do?
Nobody knows much about the males. The males that are caught are usually caught either in the nest or at light traps. And that actually nobody knows where they go or where they are, but they do find the females at some point and mate with them.
But you think like that, because I was just wondering if you can use it somehow to compare it to, if like the female does, for example, a lot more navigation or something.
Oh, I see, yeah. No, that we've never been able to do because we don't know enough about what the male does or whether it even navigates at all. They do have incredibly sexually dimorphic eyes. The visual system in the male is very different, actually. Yes, Viva?
Do they do the orienting flight every time they leave?
No, they don't. So naive bees or bees which have been placed in a new circumstance, like stuck on a stand with a ping pong ball over the front, they will come out and realise, oh my God, there's something really weird, and then they will do this flight. Maybe they'll do it twice or 3 times, and then they'll stop doing it. So subsequent nights. Yes.
Yeah, I would also have a question related to Hannah's question. Like you said that they raise their young in the stick.
Yep.
Do they do it like the domesticated bees, that it's actually only females and the males are only seasonally?
That's correct. Yeah, it's only the females that are doing this. Yeah, but there are males being raised in there too.
Okay, so, and that is all year round, because like in the domesticated bees it's only...
Yeah, it's most parts of the year, yeah.
Ah, okay.
Yeah. Okay, so you can do a really simple experiment. I'm almost embarrassed to show this experiment after all the amazing techniques we saw yesterday from the other insect lecturers. But this is a very simple experiment to demonstrate whether or not it might be the case that they use their visual system to actually look at landmarks during this orientation flight. So in this experiment, what we do, we have a stand in the forest again, but this time with only 5 nests. 4 of which are actually unoccupied, 1 of which is occupied. That's the one marked with the star, and the occupied nest is always shown with the star in every panel. Okay, so we do an experiment where we allow the bee to leave at a certain time when the light level is quite low. We're watching this with infrared goggles, by the way, and filming with an infrared camera. And we let the bee fly off, and then when the bee it makes its orientation flight, we are assuming now that it's learning that its own nest is in the middle of these other nests. In other words, it's right in the centre. It's the central nest of the 5. And if that's the case, when it comes back, it should choose that nest. So while the bee is away, we do something really evil, and we take that nest and stick it on the end of the stand and put an empty nest in the middle to find out, whether they will be tricked by this. And indeed, when they came back, they flew exactly into the central nest, precisely as predicted. 1 second later, they flew straight back out again, extraordinarily agitated. Something smelt wrong, or it felt wrong, something was wrong. And then it would continue to do kind of a flight, an orientation flight in front of these nests, and then re-enter the central nest, assuming that it had made a mistake. 1 second later, straight back out again. And it would do that over and over and over again until you took pity on the bee and placed its own nest back in the middle and the empty nest back on the end again, and then the bee entered and stayed there. It was quite happy. So that was a quite strong indication that visual landmarks were important despite the light levels. To make another experiment, equally simple, this time we put a very conspicuous white card on the end of the nest with a hole in the card, obviously, so the bee could come in and out. And this is such a conspicuous landmark that if the bee learned this landmark, then you would expect it to come back to the nest marked with that card. So while the bee is away, we leave its own nest in the middle. We don't move its nest, but we just simply take the card off its nest and stick it onto a neighbouring nest. Yes?
So in the case that you exchange those poles,
Yeah.
they never check the other poles? Like
Nope.
They don't?
Nope.
No. Okay.
The criterion for our experiments is if they land on any other nest, that's it. They've made that choice. They never do. They really rarely do that. So when you place the card on the other nest, then they return to the wrong nest, but the nest that's marked with the card that they've learnt. Yeah?
Have you ever probed how many sticks they can maximum differentiate?
No, we haven't done that experiment. You mean if we had like 50
Yes.
sticks, yeah, and just moved the...
Exactly.
We haven't done that experiment. That's an experiment we'd like to do, but except we'd like to do that in a matrix of sticks, actually. Yeah.
I noticed that the light levels here vary quite a lot. So can you, I mean, I'm more used to thinking in lux than in candela
Yeah.
per meter square. Can you tell me?
This level here is about as dim as it gets. So that's about 10 to the minus 5 lux. So it's quite dim. Yeah. So these light levels, this light level particularly, these light levels here, these return light levels are really, really dim. Yeah. You certainly can't see your hand in front of your face at those light levels. Yep. So this card experiment sort of, or was there another question? Oh, sorry. Okay. So this card experiment sort of showed that the visual system is likely used for this task. Interestingly, if you film them landing on their nests as well in very dim light, they do so with apparently no problem at all. And if you show the same film, again, this is filmed in infrared light with a high-speed camera, also at very low light levels, then if you replay this, if I come back to the next one here, replay this 10 times slower, so you can actually see how precise this landing actually is. Then they sort of observe the nest entrance and put out their legs exactly as they should and find their entrance and nest entrance and go in. It turns out that the precision of this landing is no different to that found in a day-active bee in bright daylight. We've checked this already. So these animals apparently seem to be completely unaffected by the fact that they're in light levels that are about 6 to 7, close to 8 log units dimmer than you'd find in the forest during the day. And it turns out if you stick electrodes into the photoreceptors of these bees and stimulate them with light that's equivalent to the levels of light they'd be experiencing while choosing landing on their nest or learning landmarks around the nest, it turns out that those light levels, the photoreceptors of the eye, are actually responding to single photons of light. And these single photons of light, at least in the insect literature, are sometimes referred to as bumps. So here you see the response of a megalopter photoreceptor to 5 single photons of light. There's nothing magical about this. Our photoreceptors can do this as well. But the point is that these very distinct responses were occurring in the photoreceptors of this bee while the bee was behaving and choosing landmarks and landing on its nest. That is an absurd number of photons per second, less than 5 photons per second. That's really a very small visual signal and easily swamped by noise, actually. Turns out just if you make simple calculations of signal and noise, it turns out to be, that number of photons turns out to be about 100 times dimmer, or too little at least, for megalopter to see its own nest and to land on. So it's way too little light to actually see well.
But you're assuming that they have to integrate over a certain amount of time?
Yeah, I'm coming to that. Yeah, exactly. Yeah, so in other words, there's a sort of a big gap in our knowledge. The physiology of the eyes and the way that the animal is actually able to behave in very dim light, there seems to be some gap between these two. And this is where I come to the answer to your question. And this is something now that we've been working on for a while. So our hypothesis has been that the way this gap is bridged is that in these animals, and probably this is true of most nocturnal animals, both invertebrate and vertebrate, that the visual system needs to sum photons of light in time and space. And by summing in time, it's a little bit analogous to a camera. We're having a camera and trying to photograph in dim light and increasing the exposure time on the camera to allow more light to reach the image plane. Most of you have automatic cameras these days. You've probably never done this, but old farts like me at least have actually had a camera where you can do this manually. So you can make the exposure time of the eye longer, That lets the image become brighter, but the problem that you get as a result, you cannot avoid this, is that everything that moves rapidly in the scene loses, you can't see it anymore. Okay? The longer your exposure time, the brighter the image, but everything quickly moving in the world is lost. Okay? So your temporal resolution is sacrificed. You can also sum in space. So you can take your very fine pixels in your eye, if you like, so the photoreceptors of your eye, or the single facets of a compound eye, you can think of those as being responsible for coding the information in a single visual pixel of the world, so that all of them together tile the entire world. Then one other strategy is that you could actually take a group of these pixels and pool the information together and create a kind of super pixel, if you like. And each of these much larger pixels would be seeing a much larger region of the world, and a larger region of the world contains more light. Okay, so yes, exactly, precisely, yeah. So in other words, you can make the pixel size bigger, but of course then the density of pixels goes down. The number of pixels and their density goes down, and the immediate downside of that is that you sacrifice spatial resolution. So fine spatial details in the world start to disappear when you do this. Okay, that's the downside. But if you're a nocturnal animal, it might be better to see a brighter world that's coarser and slower than to see nothing at all, which actually would have been the only alternative. Yeah?
Also the method of called super resolution, which is basically like...
You mean like hyperacuity?
No, you take 2 photos of a video and
yeah.
then just like if you're moving your camera, so
yes.
you can get like better resolution to the same pixels.
Possibly. I have no idea whether these insects are doing something like that, but it's not beyond the realm of possibility. But we haven't detected it, at least. But on the other hand, the kind of experiments we do wouldn't allow you to actually detect it. So, yeah, I can't say if that happens or not.
Actually, that is, so Gabby Newman has data suggesting that at least in the fly...
Yeah, with these retinal tremors, you mean? Yeah. Yeah, but that's the only insect I know of that does that.
So what's the story there? They move their eyes to kind of, even when they're saccading, they can kind of do this little thing, jitter their eyes, and Nico Usola has this live paper suggesting there's maybe hyperacuity through that as well. Yeah, that's right.
But that's the only insect I know that does that. They are able to vibrate the entire retina sort of behind the hard cornea of the eye. Okay, so the predictions are, and I'm only telling you this because we've been investigating this for a long time, and this probably is the basis for why these bees are able to navigate as they do at night. As light levels become lower, the predictions you'd make is that visual pixel size, according to this summation idea, would increase, the visual exposure time would also increase, would lengthen, and the visual signal to noise ratio would improve, at least for the coarser and slower visual features. Yeah?
One question and one comment. First a question. Is the spatial resolution still high enough to see the nest entrance? Or have you ever measured or computed the spatial resolution that they have?
Yeah, we have. It's not huge, but it's enough when the bee gets closer to actually detect the entrance. So what I think is happening is that they don't see much at all, just the coarse spatial details of the world when they're actually navigating back to their nest, for instance, because they've learned all the landmarks along the foraging route as well. And these much larger, much coarser landmarks, which would probably be seen reasonably well, sort of guide them almost like they're on a railway track with this sort of snapshot recall idea we talked about in an earlier lecture, until they're basically forced to come near their nest, and then they start to see the landmarks that they've learned earlier around their nest, and eventually then when they get close enough, this entrance is as big as they are, and then they can see it.
Yeah, and regarding these tiny movements of these pseudopupil, I mean, the other day you said that they have around the same compound eye as a visual, or they diurnal insect. So, and it's interesting to mention that we also tethered some wasps in Woodsole, and we got some indication that they also show tiny pseudopupil movements.
Oh,
so
okay.
maybe it's worth also to look at those.
I've never seen them in this bee, but we've used optical methods on them before, and I haven't seen it in this bee. Okay, so those are the predictions of what you'd expect if they're employing summation to see well in dim light. So long ago, I made a model that we eventually had a collaboration with Toyota, actually, that was interested in this, to see whether these kind of summation strategies that you could model for insects could actually be used to improve vision in dim light at all. And so there were a couple of mathematicians here, Henrik Malm and Oskar Magnusson, who actually turned this model that I made of this system into an algorithm that could take images or films collected in extremely dim light and then sort of actually run them through this summation algorithm and see what comes out. So this was actually done with, it's probably the most fun I've ever had with research money. Actually, we went to a very expensive German train set shop and bought all these funny buildings and the railway track. And Henrik had a ball making this. And then he got a machine vision camera which he stuck on the roof of the train. Then he ran it through this environment with all these little lights on in the houses. I mean, people who make these train sets are completely crazy. They want them to be as realistic as possible. So that was good for us. And that was the input. The very little information there, and that was the output from the algorithm. And as you see, lots of things you don't see here, you do see here. And so even though the world is a bit more sluggish, it's a little coarser, you actually, if you do the summation the right way, you maintain colour. And you see details, like Charlotte, our pet spider, walking through this scene when you didn't see it in the original video. So what this told us was that this wasn't probably such a dumb idea. Then if insects did this as well, neurally, then it would be possible for them to see a reasonably good world, a reasonably reliable visual world. As far as we know, Toyota are actually still developing this now for camera systems in cars. So we think that might have been the gap. And now I just want to quickly mention that for some years we've been trying to work out whether the visual system is actually doing this or not. It's all very well to have a model, of course, and create an algorithm that seems to be doing this. And what we wanted to do was to then try and determine whether the visual systems of nocturnal insects really do this. And to do this, we decided to turn to a more easily obtainable animal, which are hawk moths in our case, and actually start to investigate the visual system of hawk moths to find out whether we could detect this spatial and temporal summation going on inside the visual system to explain this nocturnal visual behaviour and this ability for nocturnal insects to navigate. And I'm not going to go into this in any detail because this is a whole talk in itself, but basically what we did is that we actually studied the motion processing pathways of the optic lobe, particularly in this region, which is known as the lobular plate, which I'll show you here. No, I won't. I'll just go one step forward. I'll show you where the optic lobe actually is. So this is the optic lobe. That's the central brain. So there's the central complex that you heard about yesterday from Vivek and Stanley. And in the optic lobe here, there's a region. Where is it? Here, this little orange region here. This is called the lobular plate. And in this part of the optic lobe of insects, you find all of the specialized cells for analyzing optic flow. So when an insect moves through the world and has to negotiate obstacles in flight, for instance, it's all of these very, very big cells actually in this part of the brain which are analyzing that optic flow, and that information is then used to control course during flight. Okay, so if there's an obstacle coming, there'll be cells that will realize that there's an obstacle coming and cause the animal to change direction in order to avoid hitting that obstacle. So I'm not going to, as I say, I'm not going to go into this in any detail, but what we did basically is that we stuck an electrode into this part of the brain. So here is the head of the moth. And when I say we, I should say that this was actually the work of a really talented PhD student, Anna Sturkel, who finished last year. So this is her work I'm telling you about now. It's really fantastic work she did. And basically what she did is she stuck an electrode into the brain of the hawk moth, and that hawk moth then viewed a screen where we could display moving so-called optic flow stimuli. In this case, striped patterns, sinusoidally striped patterns, which we could move left or right or up or down, and then record the responses of those cells at dimmer and dimmer light levels using filters in front of the computer monitor that made it down to starlight levels of intensity actually. And here's such a response from one such cell at starlight levels of intensity, and you see in this particular cell it liked movements of this pattern upwards, got very excited by them during the stimulation, but during movement of that pattern in the opposite direction it was highly inhibited. Anyway, I'm not going to go into the details of this, as I said, but the sort of long story made short, is that we used these responses to actually probe the ability of these moths to see spatial and temporal detail in dimmer and dimmer light levels, and then infer from those responses whether or not summation was needed to account for that ability and those responses in dim light. And the conclusions, because that's all I want to mention, is that by doing experiments like this, we discovered that indeed summation was occurring, both temporal summation and spatial summation, quite substantial amounts of summation. Anybody who wants to know the details of this, I can tell you later. I don't quite have the time to do it in this talk. But the sort of end conclusion from all this was, it turns out it does exist, and it allows these moths to see dramatically better. They have a much higher signal-to-noise ratio at all light levels, from early dusk to dim starlight levels. It allows these moths to see at light levels at least 100 to 1,000 times dimmer than they would have been able to had the summation been absent. And to do this, they require both spatial and temporal summation together to improve vision. Either one of these on their own doesn't improve it as much as both together. Yes, Nakam.
So I have a question. So because of temporal summation and things like the world is more sluggish for them, do they fly slower? I mean, can you track them with a harmonic radar or something like that to see if they fly slower to compensate for this?
That's a really good question. We've been filming them, and they do tend to, bees that are flying in dimmer and dimmer light levels, for instance, we're talking about these nocturnal bees, they do tend to fly a little bit more slowly, and they become slightly more tortuous in their flight route before they enter the nest. When it comes to bumblebees, we've done other studies on bumblebees, they definitely slow down a lot as light levels drop, but nowhere near. They slow down much more than the nocturnal bees. And some of the the bees that are really acquainted with their nest, they don't slow down at all. It's amazing. They just keep, at all light levels, no matter how dim, fly straight into the nest effectively. Yeah, so some do and some don't is the answer. Yes, Peter?
What's the period of the temporal summation?
You mean how long is that exposure time? In the dimmest light levels, the exposure time in this moth is about 300 milliseconds. Okay, and to compare that to a day active fly, that same integration time is 20 milliseconds. So they increase it by over a factor of 10 in the dimmest light levels. It grows slowly as it gets dimmer. Yeah. Yeah.
So is this a dynamic adaptation?
Yes.
And so can you, I don't know if this is too long or if it's too long you don't have to answer, but what is the mechanism of this dynamic adaptation, especially the spatial? The spatial
one, yeah, I didn't have time to go into that, but we've found the cells that are likely to do this in the first part of the optic lobe directly. I can actually go back and show you, in this region here. This part, the lamina, which is directly underneath the compound eye. It's the place where the photoreceptors send their inputs. Here there are cells which actually spread out and connect the photoreceptors together and do this pooling, that spatial summation of information. It seems to be the case that the extent of that summation increases as light levels go down. And Anna has got unpublished recordings from cells, from so-called lamina monopolar cells in this region of the optic lobe, which show a dynamic change in their spatial properties as light levels go down, from being highly lateral inhibitory in bright light, which is what you'd predict from sort of information theory, to becoming very broad spatially, having broad receptive fields in dim light, which is sort of agrees with the idea of them pooling in dim light. And but that's not published yet.
Yeah, that's very cool.
Yeah. Yes. Yes.
Could it be, I mean, I guess probably light level is more likely, but could it be that this dynamic thing is controlled by signal-to-noise ratio?
It could be, yeah. Plus, in flies, for instance, it's been shown, I think in Drosophila actually, that there's a circadian rhythm in the number and types of synapses that occur between the LMC cells in the lamina. So from day to night, it changes. So the whole circuitry seems to change in a circadian fashion.
So could you do this experiment where you keep a light level constant but change the signal-to-noise ratio?
You might. We've not done that, but that's an interesting idea. Yeah.
Just quickly, this adaptation is faster than, like you see the adaptation within, I guess, minutes or something, right?
Yeah, that's right. Yeah. Yeah.
Did you find the cells for all orientations of visual flow, and in the same ratio?
You mean these ones that are here, or you mean the motion sensitive cells?
Yeah.
Yeah, no, there's cells for every orientation of motion. Yeah, there are some that are most sensitive to upward movements, others which are really sensitive to downward movements, some are sensitive to kind of movements going this way, others to movements going this way. So really the kinds of cells that probably feed into the cells that Stanley told you about in the central complex. That have the same properties.
Ratio? Or?
I think they're fairly evenly ratioed, yes, but I don't recall if that's absolutely true. But I think they're reasonably common, all of them. Okay, so the possibly the explanation for this gap I mentioned before then are these summation strategies. But nonetheless, I mean, it still seems to me, as I said earlier, really remarkable that this bee is able to do the things that it's able to do at those light levels, because it's perishingly dark in the rainforest at night. And with those tiny eyes and a brain that has a volume less than a grain of rice, they seem to manage navigational tasks, which are truly remarkable, I have to say. So as I say, I'm not sure that we're really at the bottom of it yet, and we've worked out all of the strategies that they're able to use to perform this navigation at night. Okay, right, my second case study, unless there's more questions about the bees, etc. Yeah?
One more. Does the spatial acuity decreases with this light?
With light levels? You mean of the bees? We haven't specifically tested that, actually. But these are behavioural experiments that we've got planned to do. Yeah. Theoretically, it should. Okay, so this second, we go from now medium-range navigation at night to long-range navigation at night. This is the Australian bogan moth, Rodas infusa, which is an iconic insect in Australia. All Australians know about this insect because they, well, firstly, they annoy people a lot, especially in the capital, Canberra, because some years they actually invade Canberra because they get blown a little bit off course and get attracted by the lights of Canberra, block up buildings, close down the parliament. I mean, all sorts of things which I think are quite funny, but they're not appreciated by everybody. The Australian Aboriginals also knew about them because, as I'll show you in a moment, they end up in huge numbers in very isolated caves on the very tops of our highest mountains in Australia. And the Aborigines have known this for thousands of years and have come up from both sides of the range to actually feast on these moths and stay up there for a couple of months actually and get fat on moths, do tribal business, arrange marriages, increase the gene pool, no doubt. And then after the summer is over, they return to their tribal areas. That continued well into European occupation of Australia, but it died out, I think, in the 1860s or 70s. Anyway, this moth is interesting because it has, a little bit like the monarch butterfly, a quite remarkable migratory ability. So these moths, in the spring, they hatch, well, they develop during the winter, and the larvae eat the roots of plants, and when they pupate in the soil, and then in the spring they emerge as adults, and when they emerge, they fly from these various locations in these more arid areas of Western New South Wales and Northern New South Wales and Southern Queensland and Western Victoria. They all arrive in a very small region of our highest mountains just south of Canberra. This is the Australian Capital Territory where the capital Canberra is. And they all go to this very restricted location. And this is the kind of place they arrive at. Oh, sorry.
What's the dashed line?
Yes, I should have explained that. They've even been found in Tasmania, which is this island down here, but it's thought that they've been blown there by wind. They've not deliberately arrived there. There's no indication that they've actually actively migrated there. Yeah?
Are there actually also other mountains along the west coast?
Yes, all along, and in fact, from here all the way up to here, and they're quite tall.
For the ones coming from north, it's not the closest.
No, it isn't. But then, that's right. There are quite high mountains here, for instance, and along here. And indeed, some of them here are even slightly alpine, but there are no moths there. We've looked many times, actually. They're all congregated here. And when they get there, this is what they encounter. So these are sort of large piles of big boulders, basically, left there during the last glaciation. And it's in these piles of rocks, in cracks and crevices, that the moths arrive, and not in every crack and crevice or every little hollow. They're not really true caves in the way you might imagine. It's more like a great big area of air that's sort of enclosed within big piles of boulders, and they find their way in. And they are very isolated. There's just a conglomeration of moths in one place and a conglomeration maybe a kilometre away in another place. And this is the kind of destination they have. And when they get there, this is the opening of such a cave here, and you can see another one there. Inside on the walls of these boulders, these cave walls, they congregate in huge numbers. There are 17,000 moths per square metre. That's an enormous number. And they stay there for about 3 or 4 months in a dormant state. And then after that 3 or 4 months, and I should point out the reason they come to these very high caves is this is actually a very cool place. So these caves are quite cool even in the summer. So after 3 or 4 months, the same individuals leave the caves and fly back to where they came from, where they were born. So they make the return journey as well, the same individual. So the life cycle is about 1 year. And the question that's interested me for a long time, well, not for a long time, actually, just a few years now, although I thought this was an interesting question when I was a PhD student, but then I moved to Sweden and that was the end of that. But my main question, the main question in our group at least, is how that remarkable migratory feat is actually achieved. How do they know the direction to fly? How do they know when to stop? Okay, and how do they find their way back again in the reverse direction? How do they do that? And to do it, to find their direction, for instance, they need some kind of navigational compass. And at least the work of Jason Chapman, who's probably the world's leading authority on insect migration, he's shown a very nice paper from 2010 where he actually released particles into the wind and then tracked them where they went. This is actually modelling really, and he discovered that they blew across the channel in the prevailing winds in this fashion and reached this far over that one single night. Whereas by releasing moths from the same location and then modelling their trajectories, he realised from these calculations that they were actually able to move in directions that allowed them to go not only further, but in another direction compared to that in the wind. So this was a strong indication that they had some kind of compass mechanism. But the nature of that compass mechanism has and still is completely unknown. What exactly is that compass that allows these moths to go in the right direction? And that's what I want to tell you for the remainder of our talk, my talk, because that's what we've been working on now for the last couple of years. And to look at this question, we used this device, which Henrik and his former postdoctoral supervisor, Barry Frost, developed, which is the so-called Maritson Frost Flight Simulator. And this is a device where you can, for instance, attach a moth, you can tether a moth inside an arena, which sits on a table. Okay, so there's the arena. It's sitting down inside the arena, this moth, and it's free to fly in any azimuthal direction. There's also, if I play that again, there's also a projector here that projects a Google Earth satellite image of the local area from a mirror to a screen on the underside of the arena. It's very, very bright here. When we're doing these experiments, this is incredibly dim. I mean, as dim as nocturnal features normally are. And every time the moth moves, there's a feedback which actually moves this moving optic flow underneath the animal from nose to tail. So every time the moth moves, the optic flow moves with it. And this feeling that there is ground moving underneath, very dim ground in the actual experiment, actually causes them to keep flying. Yes?
Yeah, so my question was, so you update the rotation, but you probably assume a constant forward speed? Yeah,
we do actually. We don't vary that. And it's actually a lot slower than what you see in that image too, in that film.
But you measured, is that a reasonable assumption?
We've tried to make it as realistic as possible based on the known velocities of moths during migration, yeah. This device also has this arena also has a little device up here called an optical encoder, which is able to continuously monitor the body axis direction of the moth every 0.2 seconds, I think it is, it's reading it out. So there's a continuous readout of the orientation of the flight of this moth relative to north. And here you see, sort of from another angle, the same thing. So there's a moth inside the arena, very bright optic flow just for the purposes of demonstration, moving as the moth inside the arena is moving as well. So it's possible then to actually record the virtual flight track, if you like, of a moth in this arena as it's flying. Okay, if it's well oriented in a particular direction, it will continue to fly more or less in that direction and not deviate very much. And such a flight might look like this. Okay, you can sometimes get a bit of a loop. So over 5 minutes, you could actually trace the track. Yes?
Do they migrate in swarms? Or?
We don't know, actually. We seem to catch them in large numbers when we do catch them with light traps. It sort of suggests that they come in large clumps.
So it's also not known if there's any swarming behavior or that they, in some social context, they're...
No, it's still unknown. Most of the questions you're going to ask have no answers because we've only just been starting to work on this. So, yes?
But at least when we catch them, we have these big, bright lights going up into the sky, and my impression, at least, is that you see single moths going through.
Yes, you do. Yeah.
Groups, and so I would think that there are many in the sky, but it seems to me that they are rather going by themselves.
By themselves. On the whole, I think they are. I don't think they're not sort of flying in formation or anything. Yeah. So when you have a track like this, then you can actually you can sort of assign then a vector, which we do to this. And I'm showing you this because I'm going to show you data that uses these vectors in a moment. So that's a fairly well-directed looking flight with a reasonably long vector. But you can also get not so well-directed flights occasionally if the animal is confused or for whatever reason, experimentally manipulated reason or otherwise, then it might have a much shorter vector. The vector, of course, has not only a length, it has some direction relative to north. So that's closer to north, obviously, than that one. So in the first experiments we did a few years ago, we actually decided we wanted to find out what the natural migratory directions of the animals are in spring, that's in the forward migration, and in autumn during the return migration after they've been in the mountains. And for this purpose, we actually chose two locations. One in northern Australia, a really nice area called Mount Caputar, which is near a town called Narrabri, which you might see on a slide in a moment. There's Sydney, capital of New South Wales. This place here is called Adaminaby, and the reason why we go there is my family have had a place here for some time, and we actually use this as a field station now. So it's very nice to be able to go there and work there. Andrea and Jesse both know the place, and I'll come back to that in a moment. So we worked here as well. And what we did then, and by the way, these are the area in New South Wales, at least, where the caves are found. Above 1,500 metres, it's actually a very restricted region, shown in white there. So if you now look at the natural flight directions of moths, okay, at these two locations, so it's Narrabri and Adaminaby, Narrabri being near Mount Caputar, the orange vector shows the southward spring migration, and these grey vectors show the possible return directions, because we caught these moths in the mountains near Adaminaby, this town, and they could have been going back anywhere because they were going from their destination back home again. They could have been going anywhere. So the moths, at least up in Narrabri, these were 18 moths, and these are the individual vectors, like I showed you in the previous slide, for those 18 moths. This is a kind of a grand mean vector which weights the lengths and directions of each of the individual vectors of these moths. Okay, and this is the average direction here. And so they're going roughly in the right direction, sort of south, slightly southwest. These dashed lines here are levels of significance. So if this red vector crosses the inner dashed line, it has a significance level of p is less than 0.05. In other words, it's a 95% confidence limit. This second dashed line is 0.01, a confidence limit, so 99%. And this outer one is 0.001. Confidence limit. So in other words, with 99.9% likelihood, that is the direction which represents that population. So that looked like they were doing roughly the right thing. And then in the return migration, we had no a priori reason to believe that they would be going back to Narrabri, or back through Narrabri, in fact, to southern Queensland, because they were going in many different directions. But nonetheless, the population that we tested of, in this case, 36 moths, were mostly heading backwards in a roughly slightly northwesterly sort of direction. again with reasonably high significance. So in other words, these moths do roughly what they should, just in an apparatus like I showed you. So they've got the arena sitting on the table, we put the moth on the tether. Oh yes, sorry.
What does the length of the one?
The red one?
Yeah, I mean the length of the orange or blue line codes for?
You mean these lines here?
Why is some longer and shorter?
Oh, that's for the reason I mentioned before. That's because some moths are more oriented and some moths are less oriented. So the shorter vectors indicate a less oriented moth, so a more confused moth, if you like. So the longer ones indicate a more directed moth. Okay, so you can see that there are some short ones there, actually, but most of them are reasonably well directed. Okay, so they seem to know what direction to go in. So these moths, these moths here, we probably didn't catch the same moth, of course, but we wouldn't have caught the same moth, but nonetheless, these would be the same individuals going in this direction in spring and in this direction in autumn. So they've got a mechanism somehow of being able to switch directions in their head. So what cues might they be using to actually work out the direction that they need to go, how to follow this inherited migratory direction? The moon and the stars, we've talked about that in earlier lectures. They're relatively constant visual cues for short-range navigation. It lasts around 10 minutes, like in a dung beetle, for instance. So for the types of navigation, or orientation tasks, I should say, that Marie talked about with dung beetles, moon and the stars are perfectly good cues. They work extremely well. But if you are a long-distance navigator that migrates all night, like these moths probably do, then these cues move considerably during the night, and over a long period of time, if you're migrating over weeks, then some days the moon may not even be up, and the stars, if you can see them, probably not extremely reliable cues either all night. So the question is, what cues might they be able to use? And of course, the one that came to mind immediately, because it seems to be the cue that's used by another well-known nocturnal navigator, and these are songbirds, is the Earth's magnetic field. So we started to look then carefully, doing experiments now to see whether we could actually detect any ability of these moths to orient and in this flight arena using imposed Earth strength magnetic field. I'll point out now that, oh well, actually I won't point it out yet, I'll point it out in a second. So in other words, our question was, do bogan moths, like nocturnally migrating birds, use a magnetic compass? That was our question. And so this is a collaboration, as you might have heard. Henrik is involved in this very heavily, as is indeed Barry Frost. This is my postdoc David Dreyer, who was actually a PhD with Henrik a few years ago and now works in Lund with us. And here is a setup that we've been using now for a few years, where we have an arena sitting on a table as normal, exactly the same as before, but this time we've got a set of magnetic coils around the arena, which allows us to manipulate the Earth's magnetic field. So this is the same strength and inclination as the surrounding field, but now with these coils we have the possibility of being able to turn the azimuth of the field in any direction we want. Now, when we started these experiments, and Henrik was there as well, so he's experienced all this, we just sort of believed that the moths, if they had a magnetic sense and could use it as a compass mechanism, that they would simply turn in the magnetic field like has been found in emelin funnels with birds, because they seem to react to a turn in the field immediately, more or less, when they're at least in the mood for migrating. And for at least 2 years, we tried the same thing over and over. And here's actually us up at Mount Capitar with these coils, actually, or near Mount Capitar. There's Barry and Henrik out in the field. Yes, unfortunately, as Henrik will remember, our first results were very discouraging. So if the moths turned at all, they turned in a fashion that didn't seem to logically agree with the way the field was turned. Many moths just ignored the field entirely and just kept on going in the direction they'd been going before. There was, after at least 2 seasons, Henrik, I don't recall if I think it was 2 seasons, and much hair pulling, which in your case wasn't easy. Sorry, I had to get you back. We realised that we were doing something wrong. So this turned out to be a kind of a point where we had to sit down and think, either they don't have a magnetic compass or we're doing something fundamentally wrong. And the thing that we did, which turned out to be fundamentally wrong, of course, we realised very rapidly after thinking about it, was this isn't the only cue that the animal has in this arena. I mean, this is a horribly obvious visual cue seen above the top of the arena. The arm of the encoder is visible. There are probably wobbles and bumps inside the arena. Lots of visual cues are there as well, okay? So when we turned the magnetic field in this arrangement with these coils by, in this case, we're moving at 120 degrees, all the visual cues just stayed where they were, okay? And if they were more interested in visual cues than magnetic fields, and who knows, they may have been, then they may have just ignored the magnetic field, and at best, maybe they reacted in a completely bizarre way to the magnetic field. So that's when we started to try and get a little bit wiser, and we instead created another type of stimulus system. So we still had our arena. We tried to make the internal wall as featureless as possible and then impose two extremely distinct visual cues which would override anything that was left. And so what we did is we placed a horizon in the bottom third of the arena, and then we had a small kind of mountain top, if you like, that we put inside the arena that we could remove. That was actually connected by a very thin piece of tracing paper, very, very almost invisible tracing paper to the outside here, so we could flip it in and flip it out as we wanted. We also, it turns out, had another one down here at 120 degrees, which we could do the same thing. And in addition, we had a disc of diffuser paper that was sitting on the shaft above the animal that we could rotate above the animal as well. And that actual diffuser paper blocked out everything above the arena that could have been a visual landmark. So it had a size that was exactly right to block out everything in that above the arena. So all the coils disappeared, the encoder arm disappeared, there was nothing to be seen. The only thing to be seen was this black stripe which we put on the arena, because we thought if the moth is using visual cues, we have no actual a priori knowledge of whether those cues are in the frontal or lateral visual field or even above, because they've got extensive dorsal visual fields as well. So we created a dorsal landmark which we pointed towards the mountaintop here. And if we moved the mountaintop, we also rotated the stripe and moved it as well. Then in addition, of course, we still had the magnetic field, an earth strength magnetic field here. And then we did the following experiment when we had this type of stimulus arrangement. So we did a 5-phase experiment. So that's the kind of stimulus arrangement I just showed you. So these 5 phases each last for 5 minutes. In other words, these moths are required to fly for 25 minutes through 5 different experimental phases. The first phase, we arranged the magnetic field to be aligned with magnetic north. So this is an earth strength magnetic field, just to remind you again. And we had the 2 visual landmarks aligned eastwards at 60 degrees. The reason why we didn't line them up, though we have done this with lined up cues as well, the reason why we didn't want to line them up is we didn't know whether one of these cues would dominate over the other, and it would be very clear if we separated them if that was the case. In the second phase, all we did in the second 5 minutes is we switched both the position of the field by 120 degrees, so it now pointed sort of roughly southeast, and we also flipped the landmark out here and flipped in a landmark here. So the mountaintop was now at south. So that this whole thing, in other words, just rotated by 120 degrees for the second phase. Then what we did is make a cue conflict in phase C, and this is phase C1 here. So we left the landmarks where they were and now flipped the magnetic field back to where it was in phase A, okay? So it went back to north again. And so that was a kind of a cue conflict. So if they'd learned to associate the visual and the magnetic cues, then by phase C, 10 minutes into the experiment, this would be experienced as a cue conflict situation. To make sure that we picked up any effect properly, we repeated the phase a second time. So in other words, nothing has changed here stimulus-wise, it's just been repeated for a further 5 minutes. And then as a safety check, in the 5th phase, phase D here, we simply return to the conditions of phase A. Okay, so it's exactly the same stimulus conditions as a kind of a safety. And this is the results that we got. So again, it's exactly the same kind of plot you saw earlier here. And what you see immediately is that the moths are pretty much aligned near the landmark. So they quite like the landmark. The visual landmark is actually probably dominating over the visual field. And then if you flip everything by 120 degrees, the moths too flip by the same number of degrees. So they begin to migrate sort of almost southwards. But when the Q conflict was introduced, the directedness, as seen by the length of this vector here, this average vector, started to decrease and became insignificant. So it fell below the 95% line and became even worse during the second 5 minutes of this conflict. When we put the stimulation back to where it was in A, everything went back to normal again, and they were again reasonably oriented. Quite well oriented actually, back to where they roughly were at the beginning of the experiment. And just to control for the, whether or not this is just due to fatigue or whether there really truly is a Q conflict, showing that they must be able to detect the magnetic field, you can do the following kind of control. So basically you do exactly the same experiment, but this time in phase, instead of doing phase C1, you just keep repeating phase B. Okay? So the only difference between this phase B1, and this phase B2, compared to C1 and C2, is that the magnetic field is in a different position. Okay? It's in the position it was in phase B. That's the only difference. So the dominant landmark, which it seems to love to fly towards, this is the Q that it seems to be most attracted to, that dominant landmark is in the same place. And again, phase D is just as it was in phase A. So that's the only difference between those two experiments. And it turns out that the moths remain very, very oriented in all of these conditions. So it seems almost as if during this Q conflict, this landmark, which the moths could see very clearly and orient towards very rapidly and without any problem, seems to become invisible somehow when we flip the field back to the northward position. That's not the case if we simply leave the field where it was. So this Q conflict then seems to indicate that not only do the moths have a magnetic sense, but they seem to have somehow correlated the magnetic cue with the visual cue, and when those two become uncorrelated, then they're unable to orient. Yeah?
Why do the moths orientate towards the landmark and not their migration direction? Or was the landmark?
That's a very good question. And I think the reason for that is that this is a very unnatural experiment, actually. These visual landmarks are incredibly dominating. So we're planning to do experiments with natural panoramas to do the same kind of experiment, so there would be no tendency to be attracted to a single very dominant feature. But we know from other insects that are sort of exposed to an arena like this with a very dominant landmark, like a vertical stripe, that they can be very attracted to that. Yeah, but we used it to our advantage in this case.
Yeah, but in the experiments before, so the coil itself is not a dominant landmark, but still it makes them to keep the migration direction. And here you introduced something which is more important, kind of.
Yep. Well, much more salient, at least. So you mean the natural directions, when we just looked at their natural directions. There was no coil in those experiments, though. They were just sitting on a table in an open arena looking at the sky. Yeah, they would have seen the arm going across the top of it. Yeah, but even if that wasn't there, they would probably still have just simply oriented in the migratory direction. You can sort of do this, you can do the same experiment again, but with different starting conditions, but you get the same, oh, sorry, yeah.
Have you tried making the visual landmark unreliable, like moving the visual landmark continuously?
No, nothing like that yet, yeah. Good point though, good idea, yeah. Yes.
I'm also wondering, why do you get this population shift in their angle, in their prominent direction that they go? So when you go back to the slide and compare experiment 1, the control,
yeah,
the population direction that they go is shifted.
Oh yes, you mean that direction there isn't the same. You know, that's a very good point. Remember, we've obviously not done these necessarily on the same population of moths either. And it could be that these ones are going back to a different place than this group of moths did. So we caught them in batches and then we ran them. So yeah, so I don't know why in that particular control experiment. These control experiments were done a lot later than these experiments in the season. So it could have been that those particular moths...
Because it's interesting that internally, according to the population per experiment, that you get a less variability than between the experiments.
Right, yeah. Yeah, no, that's an interesting point. But nonetheless, the fact is that they are highly oriented in every one of these phases, nonetheless, even though the studying conditions look different. Yeah.
Can I just ask you what your p-values refer to? Is that v-test or a rally test? Or?
It's actually a so-called Moore's modified rally test. Yeah, and the Moore's modified test is like a regular rally test, only accounts for the directions, but doesn't account for how directed the animal was. So, in other words, in a rally test, you just have dots all around the outside here and they all have equal weight. Whereas the Moore's modified test not only accounts for those directions, but also how directed the animal actually was. And we've got that luxury because we've been tracking the whole track. Yeah. Yes?
Have you tried with just the magnetic cue, like no visual cue at all? Will they align?
You mean, well, that's what we tried at the start, but we have not yet done that experiment. But that's a very difficult experiment to make an arena absolutely visual cue free. That's very difficult, especially because insects have incredibly good contrast sensitivity. So you have to get this right, and it's arguable actually whether you ever could make such an arena, to be honest. But we would like to try, definitely. We're talking about that at the moment. Yeah.
You could blind them, no?
I'm sorry?
You could blind them, no?
Well, you could. I suppose you could blind them. Or you could just have them in pitch darkness. And if it's a cryptochrome mechanism, then you'd knock out the magnetic sense as well. And we're going to do those experiments this coming season. Actually, try this in the dark, and see if this works. Yes, Hannah.
So I'm not sure if this is like real or something issue, but it does look to me like in your control condition, you get this bimodal distribution that seems to get stronger over trials, right? And it sort of looks like one stays with the
You mean that and this sort of thing?
Yeah.
Yeah.
Do you think this is real? Like they're kind of choosing one to
I'm not sure. Yeah, that's the most bimodal. Well, yeah, I suppose that's slightly bimodal.
Yeah, it's more in this blue condition where this seems to happen.
Yes, yes. No, I don't know. That's a good question. I'm not sure if that has any meaning or not. Yes, anyway, as I was saying, you can do this experiment again, but you can start in other ways. So here we started instead with the magnetic field. The visual landmark's in the same place, but the magnetic field now pointed at 120. And then instead of flipping clockwise, you can flip everything 120 degrees anti-clockwise. But again, you can then introduce a cue conflict exactly as before. And again, the result is the same. So in other words, this is a repeat basically of the first experiment. Now, the interesting thing is, actually, and this is what I want to finish with, is this kind of confusion, how this actually occurs. So going from phase B to phase C1 to phase C2, you see a decline in the directedness of the population of moths. So they start from being quite directed to less directed to really undirected. And this amount of directedness is actually encoded by the so-called R star value of this Moore's modified vector that results from this weighting procedure I just mentioned. It turns out that, according to these statistics, the population is significantly directed if that R star value is greater than a value of almost 1. It's like 1.001, and it depends on the number of moths, in this case, that take part, but it's around 1. And that's actually, when it becomes greater than that value, the vector just crosses the inner dashed circle in these plots. Okay, so what we then did was to look at how R star changed in that transition from phase B to phase C1 to phase C2. And the way we did that is that we took the 5 minutes of flight in each phase, and instead of creating an average vector for the entire 5 minutes, we broke up that flight into 30-second bins and looked at the average R star value for each 30-second bin and also the average direction of that vector. So the direction and the directedness, if you like, of the vector in each 30-second bin. And then when you do that, you get a plot like this for each of the two experiments and that control I showed you. And this is shown now in phase, this is a function of time here, that's 15 minutes. So that's phase B, where it's blue, phase C1, phase C2. That dashed line there is the value of R starred, above which the flight is significantly directed and below which it isn't. And what you can see here is during phase B, in basically all the experiments, with a bit of an exception here, the control and experiment 1 and experiment 2. During phase B, the flight is highly significantly directed because it has an R* value well above 1. And when we switch now to the next phase, phase C1 and phase C1 in the 2 experiments, this is MFS, is magnetic field shift, then you see that that R* value does start to decline slowly and eventually starts to decline quite rapidly and stays quite low, below 1. The same in the experiment 2, the same thing happens. It quite quickly actually declines below 1 and stays there. The control, in contrast, stays high for the entire experiment. So phase B1 and phase B2 in the control, they stay directed the whole time and highly directed according to that R* value. So you can sort of define, if you like, a type of time to confusion. So we define that as the time at which that R* value permanently dropped below 1. And that occurred about 3.5 minutes into C1 in experiment 1 and about 1.5 minutes into phase C1 in experiment 2. Of course, it didn't occur at all in the control. So that sort of indicated roughly sort of 2 to 3 minutes it took for the moth to become completely confused and lose its orientation. So it's almost as though the moth stays oriented and it takes them a couple of minutes to realise that, oh no, these cues which I've sort of aligned or at least correlated together have become uncorrelated. And that 2-minute period elapses before that becomes obvious, and then they become completely confused and start to spiral in many cases. You can also look at the directions of the vectors in those 30-second bins, and here is, you can see that shown, time now is shown along the radius of these circles, and the direction of the vector, or the direction of the moths in those 30-second bins shown around the outside here, 0 to 360 degrees. And here is time, as I said, so 5 minutes, 10 minutes, 15 minutes as we go from B to C1 to C2. And you can see that in phase B, all of the moths in both the 2 experiments and also in the control have a direction which is 0 degrees. Now 0 degrees here is defined as the direction or the difference of the direction that it had in phase B. So obviously it was mostly in the average direction of phase B during phase B, as you'd expect, at 0 degrees. But as soon as we enter phase C1, that direction starts to deviate quite significantly. By the time we get to C2, it's deviated incredibly much. So yes, Hannah?
Maybe I missed it. These are averages of the segments of individual moths?
Yes, well, no, the population of moths. So we have the vector for the population, that red vector, and that's for 5 minutes. So what we've done now is looked at what the vector would have been in 30-second bins. So in other words, 10 30-second bins in 5 minutes.
But it's double average time and individual then?
Yes, yeah, that's right. We've done this Moore's modified averaging for each of those. That's correct, yeah. So what you see here is that the direction of those individual 30-second vectors, if you like, slowly, well, quite rapidly at one point, actually deviate from the direction that they were going to in B. The same in experiment 2, it's quite a dramatic effect, whereas in the control they keep going in the same direction. Not only do they have highly directed flights, but they have flights directed in the same direction as well. So the indication from this is that after this kind of time to confusion is elapsed, they not only are unable to hold their original direction anymore, but they can't any longer decide the direction in which to migrate. So the compass information seems to be destroyed. Yeah?
Yeah, just the circular plot you showed where you first give the contrasting cues, both visual and magnetic. So you showed it was random essentially. I was wondering if you checked to see if it was significantly bimodal, because that distribution looked the most bimodal, and if anything that's perhaps fits better with your predictions of them being confused by these two kind of contrasting cues.
Right. Now, we haven't checked that, but that's an extremely good point. Yeah. Yes, Pauline?
And concerning the circular diagrams, so you plot the direction even though it's not directed?
In the... Sorry, say again?
So you plot the direction even though...
Yes, you mean here? Yeah, that's right. That's a kind of an artifact of this. Yeah.
That's then, I think, a bit misleading, or you are very lucky that the dots are not in the sector which your data lay before, because they could be, since they are not directed, also being there. Not significantly, of course, but the chance would be the same.
That's right. No, that's actually a valid point. But I'll show you the actual vector trajectories of that whole 5-minute segment with those 30 seconds. And you'll see that they do get smaller and smaller and smaller. They do. And of course, then you could ask whether the direction is significant. And of course, it can't possibly be. That's absolutely true. But they do follow each other quite. So yeah, but that just means that these points could be anywhere, basically.
But,
Yeah.
but it kind of suggests that there is I don't know, that the dots are somewhere else where they don't supposed to be, but actually it's just coincidence if the R value
Is so small. Yeah, no, that's a valid point, actually. Yeah.
It's good for visualization, but it might also not help you if you have another set of data where it's in the sector.
What you can say is that they're absolutely not directed anymore, and that's the main point, actually.
Yeah, but that you see in the top graphs and not so much in the
Yeah.
ones below D to F.
Yeah, yeah.
But it is still so that over time, if you do many of these experiments, you will expect it to distribute in the whole. So it's quite unusual if all of them by chance with that low vector numbers would all be in that sector. That would be very unusual.
Of course, that this picture is a bit, I mean, it helps in this situation. But you would have to have a lot of populations, because these are not individuals, right? These are...
No, it's a population. Yeah. That's right, yeah.
So is what you're saying you'd like to see error bars on those dots? Or like
You could have error bars on the dots.
It's quite good, the situation that the non-directed directions are not in the sector where the directed values were, because if it were, it would be probably very difficult to explain to an audience that it's not directed, because they could fall in this sector. So it's, I wanted just to know that I understood it correctly, that the pink dots are not directed.
No, they're completely, yeah, the fact that they have a direction, it doesn't imply that that population was going significantly in that direction, because it's significantly not going in that direction. That's the point, yeah.
But I mean, it's significant. We have to always remember in biology that we have decided that 5% significance is our significance level.
Yeah, of course, but that's what you use in this experiment.
Sure, but it means that even some of this may mean something, because of course it's not a law that just because something has a p 0.08, it doesn't mean that it's random. You cannot prove random. That is very, very difficult. So that may still mean something, or it may mean nothing. I'm just saying it's not. It doesn't have to be completely without information. It doesn't have to. But I think it is without information in this case.
Yeah, exactly. And I wanted to disentangle these two things.
Sure.
Just
yeah.
Because the dots kind of mean different things. If you take the diagrams above.
We can talk about it after the lecture, but yeah, that's a good point. Yes, sorry.
So at this point, I just wanted to clarify. So how are these averaged? I think I got a little confused. So each data point, each dot is a population, which consists of how many individuals roughly?
Well, that's actually shown in the previous graph.
Okay,
it's shown in those graphs. Okay, so it's just that red vector, which is now in an entire 5-minute phase. Okay, what we've done now is broken up that 5 minutes of flight into 30-second vectors instead, and done exactly the same analysis. Okay. Exactly the same analysis.
Got it.
Yeah.
Thank you.
Okay.
Excuse me.
Yeah. Oh, sorry. Yeah.
No, maybe I didn't perfectly understand one thing, but was not possible to create a really homogeneous environment inside this arena.
A visual environment, you mean?
Yeah, yeah. So that only the magnetic compass was the cue.
Yeah, we would love to do that, but I'm not convinced we can, I'm afraid. We can get close to it, but I don't think it's perfect. But
and what about putting many landmarks symmetrical?
Yes.
Or
We've tried that too.
Yeah.
That doesn't seem to help.
Or even an object closer to the head of the moth, so that its vision is kind of more obscure.
It's more obscure. Now, we've thought about all of these things, and the problem is getting a homogeneous enough material to put on the inside of the arena.
What about if the environment rotates just for control? If you put it as homogeneous as you can, then you shift it so that they cannot really learn on something, or maybe it could be possible to measure the orientation with respect to the magnetic field. I don't know. I just guess.
No, it's worth I don't know. That's a good point.
Is it possible perhaps to place some sort of a little cap on the line that has a little LED in there? So they don't have any visual information from the outside, but they have photos. I mean, for light-dependent magnetic reception, you only need these photos, and it doesn't matter where they come from.
Yeah.
So that could be a control for that problem. I don't know how big these moths are, but that's pretty big. Maybe it's possible.
I will talk about that afterward. That's interesting. Okay, I better be quick now and wrap up. So our hypothesis, at least, is that this strategy that they might be using, they've obviously got a magnetic sense, that's clear. So from these results suggest that they have that. But it seems as though that they seem to associate, at least in our experiments, visual landmarks with the magnetic field. And our hypothesis at the moment that we're working with is that they might actually use a visual landmark in the environment as a cue to which they actually fly once they've calibrated their direction with the magnetic field. In other words, they use their compass, their magnetic compass, to work out what direction to fly in, and they look for a visual cue near or at that direction and start using that to fly towards. Now, the landmark fidelity with the magnetic compass, at least, they seem to recheck that every few minutes just based on that time to confusion. Now, that either means that they don't use their magnetic compass for like 2 minutes and they just use it every second minute and do this every couple of minutes, or, as Peter sort of alluded to this morning, the magnetic compass might actually be quite noisy and that they need to integrate to make a measurement, and that might take 2 minutes to get a reliable reading, so that within that kind of 2 minute period it's not possible actually to even notice that you've strayed away from the visual landmark, or that the landmark has gone into conflict. So that is our kind of working hypothesis at the moment, is that they use visual landmarks together with the Earth's magnetic field to steer migration at night. And we don't know of any other insect, at least, that does something quite along those lines. It's sort of like what we do with a compass when we go hiking, is to sort of look for visual landmarks in the direction we want to go. Okay, so what we're trying to do now is investigate this in a bit more detail. I just want to finish with this. So we've just finished building a non-magnetic lab in Australia. So it's no longer your lab is not the only one in the world, Henrik, as you claimed earlier. So we've just finished building this little cabin out of non-magnetic materials, and we're putting an electrophysiology setup in there and a behavioural setup with coils. And here you can see two people you recognise, I think, Jesse and Andrea. So Andrea will be doing electrophysiology in here, trying to stimulate cells of the central complex that we'll impale with electrodes there under magnetic and visual stimulation. It's just getting built up at the moment. And Anya has even been involved, part of the collaboration with Henrik, so she's been over collecting moths as well and helping with the experiments. And she and I were the major moth collectors for at least a week at one point. So the future questions that we have is how magnetic and visual information is integrated in the brain and used to drive migratory behaviour. That's a major question that we'll address with electrophysiology, among other things. Another question is where and how is magnetic information sensed? I mean, everybody that works on magnetic sensation wants to know the answer to that question. And we don't know whether the bogan moth could be a simple enough nervous system to actually find the answer to that question. We're also interested in knowing how the inherited migratory direction is actually encoded genetically, because the moths that originate, for instance, in far western New South Wales, they have to fly more or less exactly eastwards to get to these caves, whereas moths coming from southeastern Queensland, they have to fly more or less southwest. So those two populations will have two entirely different inherited migratory directions. So we'd be very interested to know how that's actually sort of encoded genetically. We're also interested in knowing how migratory and non-migratory bogon moth morphs differ genetically. There are populations in Australia that exist in Western Australia, for instance, that don't migrate at all. So it'd be very interesting to know what the differences are there genetically. And finally, another question that we'd like to answer is whether or not individual caves occupied by moths are occupied exclusively by moths from a single geographic origin. We've got some early indications using isotope studies that that's actually the case. So if it's the case, for instance, that Western New South Wales moths have their own cave and southeastern Queensland moths have their own cave, that makes the navigational task even more sophisticated, actually. And that's something we'd like to get an answer to as well. And I'd like to thank all the people that have been involved in all of the work over the years. The nocturnal vision work, the ones in red are the ones who are here or were here. So Marie Duck has been heavily involved in all the work we've done on nocturnal vision. And then the bogon moth work, Andrea and Anya Stanley has been involved in it very heavily as well. Henrik, obviously, and Jesse. And I'm really grateful to all of the fun times we've had and all the input that you've given to this project. It's been great. And in fact, thank you very much.